In 2025, a series of new tests and scans reignited the debate around the Nazca mummies, results that some research teams describe as anomalous while others call inconclusive. These three-fingered beings, once written off as elaborate hoaxes, are again at the center of scientific scrutiny. Some labs report unusual genetic markers, while peer-reviewed papers now cautiously acknowledge data gaps instead of dismissing them outright. At the same time, questions about their chain of custody remain. Many of the specimens surfaced through grave robbing and black market handling, making independent verification more complicated. Why now, and why are similar skeletal finds emerging across the globe at this exact moment? This documentary examines the new lab data, the archaeological context, the critics, and why all of it matters. And before anything else, we need to ask, were these mummies always just a hoax, or is 2025 reshaping that story? The shift in scientific consensus. For most of the academic world, the Nazca mummies were written off long before 2025. From the moment photos of the small, three-fingered figures spread online in 2017, museums and universities across the globe dismissed them as forgeries, composites stitched together from animal bones and modern materials. Peruvian officials reinforced that perception in 2023, when customs authorities confiscated a shipment of supposed mummies at Lima Airport and found that they were nothing but wooden models assembled with glue and decorated to look ancient. The Ministry of Culture publicly concluded that at least some of the items linked to the Nazca discoveries were modern fabrications, confirming what many anthropologists had argued from the start. Against that backdrop, the mainstream academic position was firm. These were not authentic remains worth serious study. And yet the story refused to disappear. Even as official investigations highlighted forgeries, other specimens continued circulating in private collections and court hearings. By late 2023 and into 2024, a wave of new reports and presentations began stirring debate. Mexican congressional hearings featured CT scans and forensic images broadcast worldwide, fueling renewed media attention. Independent analyses, including preprints shared on ResearchGate and technical summaries, circulated by groups like Abraxas, reported data suggesting unusual skeletal structure and DNA markers that did not fit neatly into human or animal reference sets. While none of these outputs came from leading peer-review journals, they demonstrated that laboratories in different countries were engaging directly with the specimens rather than relying on second-hand dismissals. The shift was not sudden, nor was it unanimous. Most archaeologists and geneticists continued to call the evidence incomplete, emphasizing that contamination, poor provenance, and hoaxes still explained much of the controversy. But what changed between 2023 and 2025 was tone. Instead of relying solely on ridicule, some experts admitted that a handful of findings, particularly consistent radiographic features and unexplained DNA sequences, warranted cautious examination. This did not mean endorsement of sensational claims. It meant that total dismissal now carried as many risks as premature acceptance. One important marker of that transition came from individual researchers quietly revising their own positions. In early 2025, for example, at least one European geneticist who had dismissed the case years earlier publicly acknowledged that raw sequence data from certain specimens included markers he could not successfully classify within available human or animal libraries. He did not argue the bodies were non-human, nor did he embrace extraordinary conclusions. Instead, he stated that while contamination could not be ruled out, further testing was justified. Comments like these mattered because they came from figures who had previously urged colleagues not to waste their reputations even mentioning the mummies. That change in language, conceding ambiguity rather than expressing certainty, signaled a new phase of the debate. Independent analyses also expanded the discussion. Multiple labs reported running carbon tests and DNA sequencing on samples sourced from separate specimens. Results varied, sometimes contradicting one another, but enough repeated anomalies were documented to make sweeping fraud explanations less convincing. Critics were then forced to argue not only that one set of remains might have been fabricated, but that several unrelated pieces from different times and places all exhibited similar irregularities. That argument was not impossible, but it demanded more effort than before. This didn't mean a scientific consensus leaned toward authenticity. Far from it. Established journals still rarely published on the subject, and when papers or summaries appeared, they often carried disclaimers about the disputed nature of the material. Many archaeologists pointed out that without transparent excavation records or clear ownership chains, any specimen could be compromised. Yet others, particularly academics working in forensic imaging, countered that CT scans and structural depictions suggested integrated anatomy that was not easy to fake convincingly. A defining feature of this transitional moment was the vocabulary scientists began using. In 2017, the language was blunt, fraud, fake, fabrication. By 2025, the words shifted, at least in some circles, to unresolved, inconclusive, anomalous. No one was claiming proof of alien biology. Instead, the cautious admission that we don't know replaced outright condemnation. In academic culture, that subtle change was significant because it opened a narrow but meaningful space for further inquiry. The atmosphere at academic gatherings reflected this shift as well. Independent researchers presented data at conferences not devoted to fringe speculation, but to forensic pathology and bioarchaeology. While such sessions remained small compared to mainstream panels, their inclusion was notable. It meant that talking about the mummies did not automatically endanger a career. Scientists could stand on a stage, present data, and frame it as an open question. That did not legitimize the mummies as authentic, but it did legitimize the idea that they merited some level of continued review. At the same time, the provenance issue weighed heavily. For every lab report raising questions, skeptics pointed to the lack of verified excavation records and the proven existence of forgeries like those seized at the Lima airport. The stronger the anomalies appeared in one dataset, the more critics stressed the risk of tainted samples and deliberate fabrication. These clashing perspectives kept the discourse balanced between intrigue and caution, neither fully collapsing into dismissal nor embracing extraordinary conclusions. By framing the subject as contested rather than conclusively resolved, 2025 presented a different landscape than the one facing scientists in 2017. Nobody in the mainstream was claiming new species had been discovered. Instead, the conversation turned to whether the data justified sustained exploration. The gradual acknowledgement that the mummies could not be extinguished through ridicule alone marked the true shift in consensus, not acceptance, but the recognition of persistent ambiguity. That ambiguity now creates pressure in other areas of analysis. If authenticity cannot be dismissed outright, then the next logical question becomes how these specimens can be situated in time. And the one tool most often relied upon to anchor authenticity in historical context is radiocarbon dating. Whether the bodies are genuine ancient burials or clever modern fakes, their age is critical to understanding what the anomalies actually mean. Carbon dating breakthroughs. While debates over authenticity dominated headlines, one of the most consequential developments of 2025 came from a quieter corner of the investigation: carbon dating. Unlike speculation about anatomy or genetics, radiocarbon analysis provides a concrete measurement of time, and the newest results added a fresh dimension to the controversy. For years, the numbers were all over the place. Early examinations reported ranges that stretched from just a few centuries old to tens of thousands of years. Such wild variation gave skeptics solid ground to dismiss the testing as compromised by contamination or poor lab practice. The problem was not entirely unexpected. Old desert mummies are notoriously difficult to date. Degraded collagen, exposure to modern handling, and even fibers introduced with preservation materials can all mislead results. That scatter of ages left the broader scientific community unwilling to treat the data as reliable evidence. By 2025, however, several independent labs ran tests again using stricter procedures. Researchers isolated interior bone collagen, rather than relying on exposed fragments, reducing surface contamination. They applied high-precision accelerator mass spectrometry, AMS, which requires only minute amounts of material but delivers far tighter results. These adjustments aligned with best practices outlined in case summaries circulated by groups like Abraxas and Rangel Martinez, and the data they produced was much more consistent. Instead of enormous spreads across thousands of years, many samples now clustered around a narrower window, approximately 1,200 to 1,800 years before present. That finding matters because it overlaps directly with the time frame of the Nazca culture, active between about 100 BCE and 800 CE. In practice, this meant that some remains, if authentic, were preserved during the very era in which the vast geoglyphs were carved onto desert plains. Supporters of further study argue this correlation is intriguing since it rules out strictly modern fabrication for some specimens, but caution remains critical. Not all dates fall within that span, and researchers repeatedly stress that dating alone cannot prove cultural linkage. At best, the overlap suggests that if independent labs continue producing congruent dates under controlled conditions, then broader questions about potential connections to Nazca traditions become worth investigating. One widely discussed case illustrates the shift in confidence. In early 2025, a sample shipped under controlled conditions to two distant labs for blind analysis yielded strikingly similar results, both placing the specimen at about 1,400 years old. This agreement contrasted sharply with the older era of contradictory dating. While margins of error remain, the concordance across independent labs reduced the chance that those particular numbers could be brushed off as random or coincidental. For many observers, this was the first credible demonstration that at least some of the specimens may genuinely be ancient. Still, ambiguity persists. Other samples have returned dates of only a few hundred years, while some fail to produce clear results at all due to contamination or insufficient collagen. Critics highlight these disparities as evidence that radiocarbon dating of these bodies remains unreliable, or at minimum inconsistent. Proponents counter that inconsistencies are not unique to this case. Archaeologists frequently wrestle with divergent dates even in accepted excavations. The critical distinction is that in this highly contested context, every irregular result is magnified by public scrutiny, often treated as ammunition in polarized debates rather than as part of the normal scientific process. These challenges underscore why researchers emphasize better material handling and verified chain of custody. If specimens lack transparent excavation records, or if samples are drawn from remains already circulated in private markets, contamination risks increase dramatically. That is why technicians in 2025 drilled deeper into bones to access less exposed collagen, ran parallel control samples, and submitted duplicates to separate labs. These measures not only improved accuracy, but also provided cross-checks to validate methods. While not solving everything, they pointed toward a standard that could reduce the recurring charge that poor technique alone explains the anomalies. The emerging picture is not uniform, and it would be misleading to suggest that every specimen aligns within the Nazca horizon. Instead, the more precise statement is that some specimens, a subset that has now been tested under stronger protocols, do. Others remain younger or ambiguous, leaving the full collection unresolved. This mixed outcome aligns with what independent summaries have already made public. Some carbon dates cluster consistently in a pre-Columbian range, others do not. That tension is now central to the discourse. What these results imply is not proof of anything extraordinary, but a reframing of how the mummies are discussed. Early dismissal relied heavily on the idea that carbon dates were meaningless scatter. By contrast, the newer results narrow at least a part of the dataset into a plausible historical period. This does not mean scientists should leap to conclusions about non-human origins. Rather, it suggests that some bodies cannot be casually categorized as modern fabrications. They may represent authentic pre-Columbian remains, regardless of their unusual anatomy, that must now be interpreted with the same caution and rigor applied to other archaeological finds. From a cultural perspective, scholars remain divided on what such a connection might mean if confirmed. Some point out that Nazca art and iconography include geometric and abstract figures, sometimes resembling unusual beings, though such interpretations are endlessly debated. Others warn against overreach. Just because a body existed within a time frame does not mean it was central to ritual or reflected in art. This is why researchers frame dating as a foundation, not a conclusion. Only when combined with anatomical, genetic, and contextual evidence can firmer links be drawn. The role of carbon dating, then, is not to settle the debate, but to shift its boundaries. If modern forgery were the entire explanation, then ancient dates across multiple labs would be unnecessary to account for. Yet here they are, complicating the narrative of universal fraud. At the same time, the younger or ambiguous results prevent advocates from overstating the case. The truth, as of 2025, is that both sets of data coexist, forcing a more nuanced interpretation. What emerges from this scientific tension is a recalibrated conversation. Instead of asking whether all the bodies are hoaxes, the question becomes which specimens truly originate in antiquity and how many were modified or fabricated along the way. By narrowing the time frame for at least some remains, carbon dating has transformed blanket skepticism into cautious curiosity. But establishing a date still leaves the central mystery untouched. What are these bodies anatomically and biologically? Knowing approximate ages helps anchor them in historical context, but the real debate persists in their physical structure. And in 2025, it was detailed forensic imaging, scans that penetrated bone and tissue, that forced both skeptics and proponents to reconsider what was possible. Inside the forensic labs, technicians confronted a different type of evidence, hard medical imaging. In 2025, high-resolution CT scans became central to the debate, offering details that earlier equipment simply could not capture. When these scans were first shared, even scientists who had long rejected the mummies as hoaxes paused to consider what they revealed. Within the skeletal structures, radiologists identified channels consistent with vascular pathways and tendon insertions that appeared to integrate naturally into bone. Such features are difficult to replicate through crude assembly alone. To some forensic experts, this level of biological integration suggested that at least certain specimens had once functioned as living organisms. The comparison to earlier imaging is striking. In 2017 and 2018, the first CT scans of the Nazca bodies were grainy and incomplete. Their blocky resolution made interpretation nearly impossible, and critics argued that what people described as anatomical features were just artifacts of low clarity. By contrast, the new scans achieved micron-level precision. Ligaments could be traced into their attachments, vascular impressions mapped along bone, and digital 3D reconstructions rotated to examine symmetry from every angle. What once looked ambiguous now appeared far more structured. To specialists in connective tissue or skeletal integration, the seamless patterns suggested original growth rather than a patchwork of glued fragments. One example often cited by imaging specialists involves the tridactyl hands. Earlier skeptics claimed they were cobbled together by taking finger bones from different skeletons and reassembling them to create 3 elongated digits. But the 2025 scans showed vascular channels and continuous tendon insertions extending from the fingertips into the wrist, all aligned with the joint spaces. These features mirrored development in living systems and did not display obvious interruptions or unnatural seams. For supporters of further study, the scans made the hoax explanation less tidy than critics once assumed. However, the imaging evidence has always carried counter-interpretations. Some anatomists argue that what looks like unusual integration might in fact be repurposed animal morphology. Several independent studies have pointed out that parts of the cranial structure match alpaca skulls closely, and comparative overlays between CT slices of the mummies and alpaca crania support that similarity. Skeptics also cite recent proteomics reports, peptides extracted from an isolated J-type head-matched alpaca proteins instead of novel human or primate signatures. If true, this would suggest that at least some specimens are animal parts presented as something more exotic. Critics use these data points to argue that any sympathetic reading of the scans could be misleading without complementary biochemical evidence. The disagreement doesn't stop there. Forensic pathologists working with the CT datasets emphasize anatomical integration that would be extremely complex to fake convincingly. Yet critics counter that artisans could have inserted bones of juvenile animals into reshaped arrangements or used adhesives to adjust morphology. Indeed, published analyses have documented traces of modern glue associated with some of the smaller figures, lending weight to the claim that hoaxes exist within the overall collection. To explain both the apparent integration and documented fabrication, some propose that the bodies under review may not all be of the same type. Certain larger specimens may preserve authentic ancient remains, while smaller ones could be constructed replicas or outright fakes. An important element that distinguishes these 2025 investigations is the public release of imaging data. DICOM files, essentially the raw output of CT scans, have been made available through repositories such as Tridactyls.org, allowing independent specialists worldwide to reanalyze the same material. Radiologists unaffiliated with the original teams can scroll through slice by slice, measure bone density, and verify whether claims about vascular channels or joint morphology hold up. This reproducibility matters because it limits the influence of prominence or authority. Instead of debating over photographs or summaries, critics and proponents alike can review identical digital files. It is one of the clearest steps towards scientific transparency in a debate often clouded by secrecy or hearsay. For forensic specialists accustomed to distinguishing authentic remains from fabrications, such access matters more than public opinion. Their assessments often hinge on micro-level subtlety, whether tendon paths terminate cleanly, whether vascular networks match natural branching, whether bone trabeculae exhibit growth patterns or abrupt cuts. High-definition imaging gives them evidence that transcends speculation. Some have gone so far as to say that no simple workshop method could reproduce these consistencies. But others remind us that isolated examples of animal bone anatomy, such as alpaca cranial forms, can look unusual when lifted out of context and could contribute to misidentification when seen without a broader comparative framework. Within the labs, what emerges is not a consensus but a narrowing range of possibilities. On one side, careful imaging shows remarkable anatomical integration, making clumsy hoax explanations harder to sustain. On the other, proteomics and morphological comparisons imply that at least some remains derive from known animals or have been altered with modern substances. These findings are not mutually exclusive. The assemblage may contain both genuine pre-Columbian remains and manipulations introduced by collectors or traffickers. The real challenge is separating one from the other with transparent data and reproducible methods. Forensic examiners emphasize that the significance lies less in whether any one body convinces skeptics, and more in how the evidence resists being collapsed into a single claim. If all specimens were modern fakes, detailed CT integration and peptide extractions indicating ancient origin would be unnecessary. If all were authentic anomalies, glue traces and alpaca proteins would not appear. Instead, both kinds of evidence persist side by side. This is why forensic labs tread carefully. The answer may not be absolute, but fractured, part authentic, part fabricated, and entirely dependent on rigorous case-by-case study. The impact of this work extends beyond anatomy. Integration and symmetry captured in CT scans hint at underlying biological processes, but they cannot address heritage or lineage. The protomic findings do the opposite. They tie samples to known terrestrial species or suggest mixtures of different tissues. Neither avenue alone can resolve the debate, but together they establish the boundaries of what needs testing next. For imaging groups, the challenge is distinguishing genuine integration from clever imitation. For biochemists, it is refining whether unexplained sequences point to contamination, animal crossover, or something not yet classified. In the end, the forensic laboratories offer clarity only to a point. They show that some structures inside these mummies look like they grew naturally, while other samples have unmistakable links to known animals and modern handling. Both supporters and critics can find evidence that reinforces their perspective. The debate now turns on which types of data should be given the most weight, and which samples carry the least questionable chain of custody. What remains clear is that by opening CT data to global review and pairing it with emerging protomic analyses, the discussion has shifted away from hearsay and toward verifiable science. These imaging and biochemical tests don't settle the controversy, but they prepare the ground for the next stage of inquiry. And it was in that next stage, in the sequencing machines of genetic labs, that the most surprising results of 2025 began to surface. The DNA that shouldn't exist. Among all the tests carried out on the Nazca mummies in 2025, the genetic work drew the most attention. When sequencing data began to circulate, scientists and independent analysts both inside and outside Peru faced results that were not straightforward at all. Instead of a unified picture, the data showed a complicated mixture. Some specimens appeared to match human genomes strongly, while others returned results with unusually high levels of unidentified or inconsistent reads. This uneven outcome is why the genetics question quickly became known as the DNA that shouldn't exist. One of the clearest cases came from a specimen labeled Ancient 3, informally referred to as Maria. According to raw data shared through the Sequence Read Archive (SRA) and later described in technical notes distributed by the Abraxas group, over 95 to 97% of the mapped reads from the sample aligned with the human reference genome, HG19. That kind of overlap is typical for authentic ancient human remains, reinforcing the argument that at least some part of the collection is from a human or human-like individual. But when analysts turned to other samples, particularly Ancient 2 and Ancient 4, the results were strikingly different. In these cases, large fractions of the sequencing reads could not be classified at all.